In this video, we are going to discuss a question on chapter 3, which is on plasma membrane transport. In this first video, we are going to discuss on part A, which is multiple choice question. Okay, question number 1. The secondary structure of protein refers to coiling and folding of the polypeptide chain. Okay, so the secondary structure, basically it has the coiling structure, okay, the alpha helix and the beta pleated sheet, okay, of the polypeptide chains. Okay, and then number 2, what is the function of intercellular junction shown in figure 1? So basically in this figure it shows you the gap junctions, okay, that are found in animal cell. So basically the function of intercellular junction is to provide cytoplasmic channel between adjacent cell. If you look at question 1, basically this one it covers on chapter 1 actually, okay, which is on macromolecule, but never mind. You have to know the structure of protein, as protein are found as one of the component of the plasma membrane. And then intercellular junction, it covers in chapter 2, which is cell. Okay, so number 3, the role of cholesterol in a cell membrane is to regulate fluidity of the membrane. So basically cholesterol is also one of the components that are found in cell membrane. So the cholesterol are found in between the molecule of phospholipid molecule in the structure of the cell membrane. Okay, so please refer to your notes. So the presence of cholesterol is important in maintaining the fluidity of the membrane, especially when the temperature is warm and also when the temperature of the surrounding is cold. Okay, so for example, if the surrounding temperature is warm, by having cholesterol in between the phospholipid molecule, okay, in the plasma membrane, this will prevent the phospholipid molecule from moving too fast. Okay, the phospholipid molecule, as you know, it will move laterally, okay, in the structure of the membrane. So if the temperature is warm, it may cause the phospholipid molecule to move rapidly and causes the membrane to become more fluid. Okay, so in order to prevent the membrane from becoming too fluid, okay, so the presence of cholesterol will hinder or will prevent the phospholipid molecule from moving too fast. Okay, so this prevents the membrane from becoming too fluid. The second condition is when the temperature is cold. Okay, so by having cholesterol in between the phospholipid molecule, so this will prevent the phospholipid molecule from tightly packed together and prevent the membrane from becoming solid. Okay, it prevents solidification of the membrane. So in order for the membrane to function properly, it cannot be too fluid or it cannot be too it cannot solidify. Okay, so basically this the presence of cholesterol only applies for animal cell. Okay, remember that, for animal cell only. And then number 4, oxygen enters the blood from alveoli in the lungs by the process known as diffusion. Okay, so alveoli will have higher concentration of oxygen compared to the blood in the blood capillaries surrounding the alveoli. Okay, so oxygen will diffuse from the high concentration of oxygen in the alveoli into the low concentration of oxygen in the blood capillaries. Okay. Okay. Question 5. Tonicity refers to the strength of a solution in relation to osmosis. Okay, so the answer will be A. So basically osmosis involves the movement of water molecule across semi-permeable membrane. And then basically for tonicity you have 3 different kinds of solution that that causes the water molecule to move from one region to another region, which is you have the first type of solution is hypertonic solution, hypotonic solution, and also isotonic solution. Okay. Number 6, when red blood cells are placed, when red blood cells were placed in solution X and Y, the cell in solution X shriveled, while those in solution Y will swell and bursts. Okay, so solution X and Y are most probably, okay, so you look at the highlighted words that shown here. Okay, so the red blood cell will shrivel in solution X, so it means that the solution X must be hypertonic solution. Okay, so it causes the water in the red blood cell to move out and causes the red blood cell to shrink and shriveled. Okay, so obviously it cannot be distilled water, it cannot be A, it cannot be D. Okay, so this left with B and C. Okay, so swell and burst, so the solution Y must be hypotonic. Okay, so the hypotonic solution outside of the red blood cell will cause water to enter into the red blood cell by osmosis and causes the red blood cell to swell and then burst. So the answer will be B. Okay, so solution Y will be distilled water. Okay, so number 7, Sanger and Nicholson fluid mosaic model of the membrane proposed that the membrane consists of, okay, D, which is protein molecules embedded in the fluid bilayer of phospholipid. So this model is called as fluid mosaic model because from the word mosaic, okay, it refers to the presence of, for example, protein that are embedded in between, okay, the phospholipid molecules in the membrane. Okay, so the protein are embedded, okay, in between the phospholipid molecules in the membrane of in the plasma membrane, for example. Okay, so that's why it has the term mosaic here. Okay, so for fluid, okay, why is it fluid? Because the molecules, especially phospholipid, found in the membrane, they can move in two ways. Okay, which is the first movement is through lateral movement. So the phospholipid molecule will move laterally, okay, along the plane of the membrane, or so this will occur frequently. Okay, and the second movement is through flip-flop. Okay, so flip-flop movement of phospholipid molecule will occur rarely. Okay, it occurs rarely. And okay, so number 8, where would you most likely find a transmembrane protein? Okay, so transmembrane protein are membrane protein, okay, that are embedded, okay, that are embedded in the phospholipid bilayer, okay, of the membrane. So transmembrane protein are also known as integral protein. Okay, integral protein that are embedded. So therefore, the answer will be D. Okay, so the transmembrane protein will span. Okay, will span. They are found along, okay, throughout the membrane, with part of the membrane visible on both sides of both the inside and outside of the cell. Okay, so when the protein are embedded, okay, so there some of the part of the protein will protrude, okay, to the inside of the cell. Inside, it means that into facing the cytoplasm. Outside, facing the extracellular fluid. Okay, so as you can see, for example, in this node, okay, if I can find the structure of a of the membrane, okay, as you can see from this diagram here, okay, so this is integral protein or transmembrane protein. So it has basically the region that are in the core of the membrane. Okay, so this region here, and the one referred in the in the answer here, okay, visible on both sides. So inside here, kan, so ada lebihan sikit kat sini. okay, and also on the outside, okay, facing the extracellular fluid side. So this is what we call as integral protein. So there is also another type of protein found in the membrane, which is peripheral protein. Peripheral protein are not embedded, but they are found at the surface of the membrane, facing the inside of the cell, okay, facing the cytoplasm. Peripheral protein are not found on the outside, but on the inside of the membrane, okay, facing the cytoplasm. Question 9. The membranes of winter wheat, okay, which is a type of wheat plant, okay, which can remain fluid in extreme cold temperature, okay. So in order to prevent the membrane of this plant, okay, the winter wheat, from solidify, okay, due to really cold temperature, so the phospholipid molecule of the membranes, okay, has to made up of unsaturated phospholipid, as the tail of the phospholipid molecule will bend, okay, they form kings, okay, kings, due to the presence of double bond in the hydrophobic tail of the phospholipid molecule. So it causes the formation of kink, okay, that prevent the phospholipid molecule from packing close together, and this will prevent the membrane from solidify during cold temperature. Okay, so if you refer to your notes here, for example, if I can find it, okay, so this one, okay, so by having unsaturated fatty acid in the structure of phospholipid, this prevents the phospholipid molecule from packing close together and prevent the phospholipid from solidifying, okay. Okay, number 10. The sodium-potassium pump is called an electrogenic pump because it will contribute to membrane potential. Okay, so basically the function of sodium-potassium pump is to pump out 3 sodium ion out of the cell and 2 potassium ion into the cell. So you have more positive ion moving out of the cell. So this causes the outside of the cell membrane to be positively charged. So for example here, like I've drawn here, okay, so this is, for example, a cell, okay, so you have the plasma membrane. So for normal cell, okay, so usually all cell will have will be positively charged outside and negatively charged inside, okay. So when the presence of sodium potassium pump will cause 3 sodium to move out and 2 potassium to move in. So more positive charge moving out of the cell and less positive charge move into the cell, okay. So this will create a membrane potential, okay, a membrane potential difference in the charge of the plasma membrane outside and inside of the cell. Outside is positively charged, inside is negatively charged. This will create membrane potential, okay. So number 11, which substance moves into the cell using energy from ATP hydrolysis? Okay, so when we see the word ATP, we know instantly, okay, that this one involves active transport, okay, active transport, as it uses energy, okay. So if we look at the choices, obviously A will not be the answer. Movement of water is through passive transport, either through diffusion, okay, or facilitated diffusion using aquaporins. Number 4B, facilitated diffusion of sodium through sodium ion channel. Okay, so channel basically it involves passive movement, facilitated diffusion, okay. For D, okay, movement of lactose from a medium containing higher concentration of lactose, okay. So the movement of lactose from high concentration to low concentration is obviously passive transport, so it does not require any energy. So that leaves you with C, okay, which is movement of sodium ion from a region of lower concentration to a region of higher concentration. So basically, if you want to understand more on this, you have to understand on sodium potassium pump. Okay, so in order for sodium potassium pump to pump 3 sodium out, okay, so it requires ATP molecule to be hydrolyzed by releasing the one of its phosphate. Okay, so ATP stands for adenosine triphosphate. So one of the phosphate will be released and attached, okay, to the sodium potassium pump, okay, and causes the sodium potassium pump to change shape and causes the sodium to be released outside of the cell. Okay, so from low concentration to high concentration inside cell. Okay, inside cell, the concentration of sodium is low compared to the concentration of sodium outside of the cell, which is high. Okay, so the movement is from low concentration to high concentration of sodium ion. Okay, so number 12, Hydrilla is a freshwater aquatic plant often used in aquarium. Okay, so how would a Hydrilla cell be affected when placed in salt water? Okay, so you place the Hydrilla in a more saline solution, okay, more hypertonic solution. So this will cause water from the cell of the Hydrilla to diffuse out through osmosis and causes the cell of the Hydrilla, okay, to shrink. Okay, so this phenomena we call it as plasmolysis. Okay, the cell is plasmolyzed. The process is plasmolysis, where the plasma membrane will shrink away from the cell wall. Okay. Number 13. Which process enables cholesterol to enter liver cells? Okay, so cholesterol, it enters into cell through receptor mediated endocytosis. So enter, it means that from outside, enter into the cell. So endocytosis. Cholesterol will bind to receptor that are found on the plasma membrane of the liver cell. So cholesterol will bind first to the receptor protein, and the and the region that has the receptor protein will fold inward, okay, to form a vesicle. We call it as coated vesicles. Okay, coated vesicles. Okay, to bring in cholesterol from outside into the cell in large quantity. Okay, bulk transport. It is a type of bulk transport. And remember, bulk transport is actually a type of active transport. Okay, number 14. Movement of water molecules across membrane into a cell can be increased by aquaporins. Okay, so to increase the process of water, diffusion of water, okay, into cell, from outside into cell, is by letting the water to enter into the cell through the aquaporins. Okay, plasma membrane of cell, they will allow water to diffuse through the phospholipid bilayer. Okay, but the rate of diffusion of water, okay, through the plasma membrane alone without aquaporin will be slow. Okay, the diffusion of water molecule through the plasma membrane will be very slow. And to increase the rate of diffusion of water across the membrane is by having aquaporins. Okay, aquaporins will increase, okay, the movement of water across cell, okay, across the cell membrane. So aquaporin is basically a channel protein. Okay, number 15. Which of the following are example of bulk transport across the membrane? So the answer will be exocytosis and also endocytosis, which is the answer is B. Okay, so endocytosis, exocytosis, a type of bulk transport. They fall under bulk transport, which is basically bulk transport is active transport. Okay, it falls under active transport. Number 16, what happens when a cell is placed in hypotonic solution? Okay, so hypotonic solution will have a higher amount of water compared to the concentration of solute inside cell. So solute inside cell will be higher. Okay, so this will cause water from the hypotonic solution to enter into the cell. Okay, to enter into the cell towards the area of higher solute concentration in the cell. Okay, number 17, which of the following statement is not true regarding phospholipid in plasma membrane? Okay, not true. Okay, so we look at statement A. Lateral movement of phospholipid molecule in the same layer occurs frequently. So this one is correct. Phospholipid molecule can move laterally, okay, and the movement of the phospholipid is frequent. It's frequent. For C, we look at C first. Non-polar tails of the phospholipids are attracted to one another by hydrophobic interaction. So this enables, okay, the formation of the phospholipid bilayer itself, okay, through the interaction of the hydrophobic tails of the phospholipid molecule, okay, to form the bilayer of the membrane. And then D, unsaturated hydrocarbon tails of the phospholipid have kinks, okay, that enhance membrane fluidity. So this is especially important in plant, okay, as we have looked at in the winter week earlier. Okay. Okay, so B, so that's leave you with B as the incorrect statement, not true, which is flip-flop movement of the phospholipid molecule in the same layer occurs frequently. So there is two wrong facts inside this statement, which is flip-flop movement of phospholipid involves the phospholipid molecule to swap, okay, to swap place, okay, from the phospholipid at the top layer will change its place from top to bottom. Okay, the bottom layer will be at the top. Okay, so that is flip flop. So so it says here in the same layer, so that is incorrect. Okay, in the same layer is incorrect. Okay, frequent movement of flip flop is incorrect because for flip flop movement of phospholipid molecule, it will occur rarely. Okay, so if you look at in this note, okay, lateral movement will occur really frequently, really fast. Okay, as for flip flop, it occurs really rarely. Okay, so it involves the movement of the phospholipid molecule to change place, okay, from top to bottom and vice versa. Okay, so number 18, choose the correct sequence of events relating to transport of potassium ions via sodium potassium pump. Okay, so okay, so for this one, okay, the movement of sodium ion is from outside, okay, into the cell. Okay, so outside into the cell. So the answer will be D, where we are going to look at one by one. Okay, so firstly, potassium ion will bind to membrane protein. Okay, so the membrane protein involved here is of type carrier protein, okay, which is the sodium potassium pump. It is a carrier protein, okay, where the protein will expose the binding site of potassium to the extracellular fluid site. Okay, so potassium will bind to the membrane protein. Okay, so if you look at the diagram here, okay, in your notes, okay, so slide 63, as you can see here. Okay, so potassium ion from outside will bind to the membrane protein, okay, which is to the carrier, and then as you can see from this diagram here, you have the protein will have phosphate, okay, bound to the carrier. Okay, so the binding of the potassium, okay, into the binding site of the protein will cause the phosphate to be released. Okay, we call it as dephosphorylation. Okay, so this will lead to statement number 4, okay, which is membrane protein loses a phosphate group. Okay, as you can see from this diagram here. And then what happened next is that once the phosphate is released, okay, from the carrier protein or from the pump, it causes the sodium potassium pump to change shape. Okay, so which is lead to statement number 2. Shape of the membrane is restored. Okay, so it return back to the normal shape. Okay, normal shape of the sodium potassium pump, which is here. Okay, which is here. Initially, it is in this position. Okay, in this position. So therefore, once the sodium potassium pump or the protein changes shape, so now the binding site is not compatible for the potassium ion to bind anymore. So therefore, the potassium ion, okay, will release into the cell. Okay, so potassium are released into the cell. Okay, so the answer will be D. Okay, so please, if you don't understand, if you are not clear yet of how sodium potassium pump works, please read up this slide, which is slide 63. Question 19. Movement of water through a semi-permeable membrane is called as osmosis. Okay, so every time it involves the movement of water molecule across semi-permeable membrane, it is the process of osmosis. Number 20. Diffusion is movement of molecules from an area of high concentration to an area of low concentration, obviously. Okay, so number 21. A cell may transport certain substance in vesicle if the substance is too large to cross the membrane. So if the substance is too large, it cannot depend on carrier protein to transport the substance, as the substance is too large. Okay, so in order to bring in a large substance or substance in large quantity, it is through endocytosis, for example, endocytosis or phagocytosis, for example. Okay, so the process, okay, of bringing in these large molecules or a large quantity of molecules into cell involve, okay, the formation of vesicles. Okay, involve the formation of vesicle, as you can see from the notes. Okay, so as you can see from the notes, slide 73. Okay, the process of phagocytosis, okay, which is a type of endocytosis. Okay, endocytosis will bring in large molecules or large quantity of molecules into cell through the inward folding of the membrane, okay, to surround the food, for example, or the nutrient. Okay, so until it forms a food vacuole. Okay, a food vacuole, for example, vacuole or vesicle. Okay, so this will bring in the substance into the cell through the formation of vesicle. Okay, so question 22. Which solution causes water to move out of an animal cell, resulting the cell to shrivel? So obviously the answer would be hypertonic. Okay, so water from the animal cell will diffuse out, okay, through the solution through the process of osmosis. So always include the word osmosis. The movement of water is always through osmosis, okay, across the membrane. Question 23. These processes are involved in the movement of sodium ions across the cell membrane of animals, okay, except osmosis. Okay, so obviously the question asks you on the movement of sodium ions. So as I've mentioned earlier, okay, osmosis is only for the movement of water molecule. Okay, so as for sodium can enter into the cell through either active transport such as sodium potassium pump, or passive transport such as voltage gated channel for sodium, for example, that are found along the axon of neuron. Okay, so this is through passive transport, which is, to be specific, it is a type of facilitated diffusion. Okay, number 24, which of the following substances is most likely transported into a cell by receptor mediated endocytosis? So obvious example given in your notes is cholesterol. Okay, so cholesterol are taken into the cell through receptor mediated endocytosis, as the cholesterol will bind, okay, to the receptor protein that are found on the plasma membrane facing the extracellular fluid. Okay, so the cholesterol in the extracellular fluid will bind to the receptor protein and causes the membrane of the plasma membrane to fold inward and form coated vesicle to bring in cholesterol into the cell. Okay, so that's the end of part A. Okay, so I hope you understand what have been discussed. If you have question, then you can ask, okay, during the tutorial session. Okay, class, thank you.